Muchas gracias por estar por el día de hoy con nosotros. Hoy tendremos el seminario del doctor Evandro Fang. The Dr. Evandro Fang is Associate Professor of Molecular Gerontology at University of Oslo, and his group is working on the molecular mechanism of human aging and aging-predisposed neurodegeneration. More specifically, the Fang laboratory is focusing on the molecular mechanism behind of how cells clear the damaged and aged mitochondria, a process termed mitophagy, as well as the role of the Nad plus mitophagy excess in healthy aging and Alzheimer disease inhibition. Nad plus is a fundamental molecule in life and health and decreases in aging and Alzheimer disease. Dr. Fang is fascinated with and actively engaged in moving his laboratory findings to transnational applications and is involved in more than 5 NAD-based clinical trials with overarching goal of establishing novel and safety biological approach to promote longer and healthier human lives. After finishing his PhD at the Chinese University in Hong Kong, he had a 6-year postdoc training with Professor Wilmore Bohr on molecular gerontology at the National Institute of Aging in Baltimore. He opened his lab in Oslo in the fall of 2017. He has published over 100 papers in international peer-reviewed journals, including papers in Cell Metabolism, Nature Reviews, MCV, Nature Neuroscience, Nature Aging, and Nature Biomedical Engineering, among others. He has received several awards, including the Walter Williams Scholar on Aging in 2016 by NIA in USA, and the Scientific Award to Young Scientists in the Natural Science for 2020 by the Royal Norwegian Society of Science and Letters in Norway. He is the founding coordinator of the Norwegian Center on Healthy Aging Network, known as a NO-AGE, the Norwegian National Anti-Alzheimer Disease Network, and the Hong Kong Nordic Research Network. Today, he will present the seminar, Aging, Alzheimer's Disease, and Artificial Intelligence. Thank you very much, Dr. Fang, for accepting the invitation. It's a pleasure to have you here today.
Thank you so much, Professor Carmen Viva, for this wonderful invitation. And it's our first time to see each other after 7 years, so I'm very happy to see you. And today, in the coming 45 to 50 minutes, I would like to share with you a couple of stories we have been working on. Firstly, I will share with you the current understanding of aging, NAD, autophagy, mitophagy, and Alzheimer's disease. Then I will elaborate 4 stories we have been working on. The first one is aging drivers neurodegeneration via impairment of the NAD mitophagy signaling. The second project is defective mitophagy is a risk factor of Alzheimer's disease. The third one is ULK1 reduction is a driver of AD with some unpublished data. And the last one to share with you our recent artificial intelligence-based project. And finally, I will give you a conclusion. So I'm very interested in art. When I was a small kid, I've been fascinated by the portrait of Picasso. And you can see this is a picture of him at the age of 15. And the second one, after 10 years, and there's no big difference for us when we are young. However, when we're getting older, you can understand how time flies. Picasso, age 91, 1 year before his death, he had his last portrait. And it shows that the artist was so struggling of his health, loss of the hair, hearing, a huge amount of wrinkles, and an unhappy face. So with this in mind, I always ask, we needed to do the aging research, why it is timely and important. if you see the epidemiological statistics regarding the aging in the developed countries, and for the case of Norway, it just says by 2030, the average lifespan for females will be around 85 years old, while South Korea will be reached over 92 years old. This will be the similar situation for men. They can have average lifespan over 80, between 80 to 85. Why we do not like aging? Because it is arguably the primary risk factor for all the disorders you can list. For example, dementia, cardiovascular disease, and cancer. So we aim to understand the molecular mechanism of aging and then to slow down the aging process if we cannot stop it. And we aim to targeting on aging to save the incidency of many age predisposed disorders. I always ask my audience, how do you foresee your health in your 90s? I hope after my talk, you will be more optimistic than Picasso regarding the answers to this question. To find a way to slow down the aging, we need to understand the molecular mechanism of aging. Colleagues in the field has proposed the different causes of aging or hallmarks of aging, such as DNA damage, telomere attrition, epigenetic alterations, mitochondrial dysfunction, dysregulated nutrient sensing, stem cell exhaustion, cellular senescence, altered cell-to-cell, organ-to-organ communication, loss of proteostasis. So in addition to those 9 hallmarks of aging, we and others together have been proposed that compromised autophagy is an independent hallmark of aging, while there's overlap with between both of them. However, autophagy impairment can be distinguished as an independent hallmark of aging. Keep in mind, all those hallmarks of aging are intertwined together. Those are just artificial names. As a small lab we can't work on all hallmarks of aging. So then I asked whether there's one parameter that links to all the hallmarks of aging. Then we can focus on one parameter. Then that would be more feasible. And very interestingly, in 2016, we wrote a review paper and highlight the linkage of NAD+ reduction to all the hallmarks of aging. And my group is working, so with this in mind, my group have 3 research topics. The first one is the molecular mechanism of NAD reduction in the aging humans as well as animals. The second topic is age-predisposed Alzheimer's disease. We are interested in impairment of the engine, which participant as the garbage recycling car to reduce the brain garbage. And we have been actively using artificial intelligence as an approach for drug development. As a basic researcher, we cannot work in with the elderly people directly. And we have been using different model systems to address our questions. In my group, we use the roundworm C. elegans, which have a lifespan of 3 weeks. The Drosophila, they can have lifespan up to 2 months. Mice, as well as human iPSC-derived neurons for our mechanistic and drug development studies. So first, I want to share with you the background of NAD+, why it is important in aging, as well as why it is reduced during aging. To address those questions, we need to understand what is NAD, how it is produced, and what are the consumers of NAD. So NAD is a molecule available in all the cell types in our human body. A normal people like you and me, who can die in 20 seconds, estimated if without NAD production. And the right part is a study showing an age-dependent reduction of NAD+ in the human skin tissue. So I want to share with you how NAD+ was discovered. It was started by a group of German professors. After the work, they went outside for the beer, and one scientist asked, how the beer or fermentation comes. If you are a generic person, then the knowledge of sugar plus yeast to generate alcohol and CO2, that would suffice you. However, for a stringent scientist, you may want to know more. And this drives them to notice that it is a glucose to be metabolized to pyruvate. And this process won't happen until you have the NAD+ in, which has been converted to NADH, the reduced form. Pyruvate will be further converted to acetaldehyde and then alcohol. The last step, turning to be alcohol, we need the NADH to provide one H+. And in this process, it converts to NAD+ for the fermentation to continue. And this explains why the fermentation comes, it never stops. So why NAD is so important? As you may know, this is a Nobel Prize season, and the importance of NAD is credited by the awards of 4 Nobel prizes to 6 people for their purification of NAD, identification of structure. If you still recall, in our textbook, the classical understanding of NAD is its functions in glycolysis, TCA cycle, oxidase, and beta oxidation. For example, glucose uses NAD+ to convert to pyruvate, which goes into the matrix of mitochondria to participate in TCA cycle, which also uses NAD. And the fat we consumed can be digested to acetyl-CoA by the using of NAD. So in those 3 steps, NAD converted to NADH, not digested. So NADH is used by the mitochondrial oxidase complex 1 to initiate the ATP production. So that explains why NAD+ is so essential and critical for our life and health. To understand why NAD is reduced during aging, we need to know how NAD is produced. There are 3 known pathways for NAD synthesis, including the kynurenine pathway. Cells use tryptophan to generate NAD+, which is a time-consuming and tedious step, which is not so efficient. The second one is press-handler pathway. Cell uses nicotinic acid to generate NAD+, which is much more efficient. However, many individuals who are taking nicotinic acid can have side effects like flushing and itching, which limits its clinical application. The third pathway has got a lot of attention in those years. So all those NAD+ not converted but digested to nicotinamide or NAM, or named vitamin B3 in our daily life. So NAM is converted to nicotinamide mononucleotide, and finally NAD+. So this is a salvage pathway. In early 2000, Charles Brenner identified nicotinamide riboside, NR, as an independent precursor, which enables this salvage pathway and recycling. So the current understanding of NAD in healthy longevity has coined as a cofactor for different processes. For example, NAD is essential for PARPs to initiate DNA repair pathways to maintain genomic stability. NAD+ is also important for the NAD-dependent deacylase sirtuins to initiate its roles in transcriptional regulation, which regulates genes which encoding proteins participating in metabolism neuronal plasticity, health span, lifespan. However, NAD+ is also essential for CD38, CD157 functions on mitochondrial transfer, social behavior. Although when we are getting older, CD38 consumes a lot of NAD. When we are getting older, the PAPs also consumes or drives the depletion of NAD+. So how to increase cellular NAD+? One is to use nicotinamide or vitamin B3 here. We can also have the nicotinamide riboside or nicotinamide mononucleotide. And in my lab, you use different NAD precursors for the purpose of NAD+ repletion. So I want to start my first project, which started in 2012 when I was a postdoc with Professor William Ball. We were interested in a group of premature aging disorders, meaning the people, their biological age is much older than their passport age. For example, this lady, she was only 48, but looks like she was 70s or 80s. And I was focusing on their dermapigmentosum, Werner syndrome, ataxia, telangiectasia. They were majorly caused by mutation of genes involved in coding protein involved in DNA repair. So we asked, in addition to DNA damage, are there any other reasons to cause the accelerated aging process? Because when we understand such reasons, we can also apply such information to the normal aging. With this in mind, with my colleagues, we showed that those three disorders show mitochondrial clinical features, as those three disorders marked in green are clustered well with the red ones, which are clinically identified mitochondrial disease. Because this is an artificial intelligence-based method, we want to use wet lab to validate those disorders are indeed with mitochondrial dysfunction. So what we did, we collected primary and immortalized cells of XPA group, XPA from the primary cells with temporary knockdown, 2 groups, XPA cells with knockdown using SY5Y cell, and an ATM. And we checked the mitochondrial membrane potential. And you can see the red ones are the disease model. They all showed high levels of mitochondrial membrane potential. Those disorders also show increased mitochondrial content. While in normal conditions, higher mitochondrial content is good because you have more healthy mitochondria. However, in this condition, it is accumulated damaged mitochondria, and I will elaborate it further later, and increased cellular loss and mitochondrial loss. So all those data indicate mitochondrial impairment in those premature aging disorders. Then we asked what are the molecular mechanisms in addition to DNA repair deficiency. We checked proteomics, RNA-seq, as well as metabolomics. We were extremely interested in metabolic data among a total of 200 metabolites by using the mouse brain from this ataxia telangiectasia premature aging mouse model. And we saw NAD+ was already 60% reduction in the 2-month-old mice compared to that of sex and age matched wild type. When we gave NAD+ precursor NR in drinking water, we were able to instantly increase NAD+ in tissues adjacent to that of wild type. As the AT mice had short lifespan, only 3 to 5 months, while the wild type 2.5 to 3 years, we wondered whether reduced NAD can cause such premature aging phenotypes. And what we found that with the NAD+ treatment in the AT mice, we saw dramatic extension of lifespan. And because this is a premature aging disease, we also wondered whether NAD supplementation was able to improve lifespan in the normal mice. And indeed, it showed a 5 to 10% extension of lifespan, not so dramatic, but statistically different. Our data was in line with a science paper published in the same year by Johan Orvin's group, which showed that from 2-year-old, the wild-type mice treated with NR precursor showed statistically increase of the lifespan. Our aim is not only to increase the lifespan, but also the quality of life. So we wondered whether NAD+ supplementation was able to improve the memory performance in the AT mice. Using the Y-maze, which is a procedure for spontaneous alteration performance, the ATM mice showed worse score compared to Y-type. And you can see with our precursor NAD, precursor NR treatment, the benefit increased its score compatible to that Y-type. To consolidate this result, we also used another precursor NMN, and we saw similar result, which indicates the importance of NAD. Because those studies were in ataxia telangiectasia and xeroderma pigmentosum, those two premature aging disorders, and then we want to challenge ourselves whether reduced NAD+ are only causes for those two premature aging disorders, or it can be the common cause for different types of premature aging disorders. And with this in mind, we included Werner syndrome, which is a classical premature aging disease, and asked whether NAD reduction is a cause. So we show NAD+ increased lifespan in C. elegans models in the Werner syndrome. We didn't do that in mice because the mice model of Werner didn't recapitulate short lifespan feature as shown in the humans. What we did, we used Drosophila as another animal model to validate our results in C. elegans. And while the temporary knockdown of Werner in Drosophila, which reduced lifespan, with the NAD precursor NMN treatment, it extended lifespan from 44 to 56 days. And we also saw a 20% increase of lifespan in the Y type, so indicate the NAD benefit to lifespan and health span are cross-species preserved. As I showed you, NAD+ improves healthy longevity as well as neuronal protection with data I didn't show you. But what are the molecular mechanisms underneath? And based on our results, we have been focused on autophagy, especially one subtype of autophagy, that is mitophagy. We do not exclude the other pathways are also important. So what is autophagy? It is a cellular process to eliminate a garbage or for survival when we are starved or when we are doing exercise. For example, when we do exercise, AMPK is increased. When the starvation comes, mTOR is inhibited, which drives the activation of ULK1 to recruit other proteins to initiate this ULK1 initiation complex, followed by the generation of this double membrane factor, elongation of this factor, maturation, and finally to fuse with acidic lysosome to degrade the component and to recycle. And the autophagy process is very well controlled, and with many proteins being sequentially recruited for this pathway. Due to time concern, I want to go to details, but the importance of autophagy has been credited as the Nobel Prize in Physiology or Medicine in 2015 was to a Japanese professor for his contribution on the mechanistic studies of autophagy. And if this autophagy only targeting mitochondria, we call it mitophagy. So how to quantify mitophagy in cells as well as in animals? So a protein called schema, if with a mitochondrial tag to enable it to express in the matrix of mitochondria. If the mitochondria are healthy, you'll see green color, like in the pH of 8. However, when the mitochondria is undergoing mitophagy in the low pH, like 4, you will see the color changes from green to red. So for example, here is a HeLa cell with a mitochondria, and in the normal condition, you only see less than 5% of mitophagy, this red dot. However, the importance of mitophagy is evidenced by this exogenous stress, like FCCP treatment. And you saw dramatic occurrence of red dots to eliminate damaged mitochondria for the survival of the cells. So with this in mind, we checked mitophagy in the brain of the AT cerebellum region. And we saw in the AT mice, for example, compare the white condition, normally you see very beautiful and condensed crystal inside. Those are the healthy mitochondria. However, in the AT, we saw heterogeneous size of mitochondria and with increased percentage of damaged mitochondria as evidenced by loss of crystal. However, with NAD precursor NR treatment, we saw homeostasis of mitochondria, reduced damaged mitochondria, as well as increased mitophagy-like situation like this. Even, and this, our data were consolidated by using another precursor and we saw increased mitophagy. So what our data show, I want to give you a summary as we didn't have time to show the exact results. We showed that when NAD is increased, which increase the tune function to transcriptionally upregulate genes involved in mitophagy, such as PINK1, PTR1, and DCT1. They may also increase at protein level, could be independent of CT1 activity. It also has been shown that CT1 can deacylate FOXO1, FOXO3, which are transcriptionally regulator on genes involved in autophagy, as well as the involvement of many other pathways. I want to go to detail. If you're interested, you can check this paper. So here, I want to give you a summary on the first part of my talk. We wanted to propose a new unified mechanism of aging while focusing on nuclear DNA repair, mitochondrial homeostasis, and subcellular organ-to-organ communication. We propose that low mitochondrial, no DNA damage when we were young can be efficiently eliminated by DNA repair. The proteins NAD and sirtuin can also regulate DNA repair. For example, it can increase the phosphorylation of Cu70 to increase the repair of DNA double-strand break in neurons. The data I didn't show you here, but we published in our 2016 paper. The NAD sirtuin pathway can also regulate mitochondrial numbers biogenesis, increase the resilience of mitochondria against exogenous stress, as well as to reduce damaged mitochondria via induction of the mitophagy receptor NICs, among other proteins. This could be the way to increase the neuronal resilience. However, when we are getting older, if you show the animation again, so it's a huge increase of DNA damage, which can drive the depletion of NAD+, while other pathways like CD38 may also play a role. This could drive the neuronal death. So how we show NAD+ is reduction is a culprit rather than a bystander, because when we pharmacologically and genetically increase NAD+, we saw increased nuclear DNA repair, increased mitochondrial function, increased homeostasis of subcellular organs. And this may contribute to healthy longevity in those premature aging, as well as normal animal models. Definitely, the NAD sirtuin pathway can also participate in many other pathways, as reported by colleagues in the field. As I mentioned to you, while working on the premature aging, we showed NAD reduction and defective mitophagy are drivers of accelerated aging. We wondered whether those molecular mechanisms are also applied to normal aging, as well as normal age predisposed disease like Alzheimer's disease. Why we are interested in Alzheimer's disease? Because Alzheimer's disease constitutes 70% of dementia, which was proposed to be 50 million in the world, with the number to be increased to 82 in the coming 80 years. And it caused formidable socio-economic pressure. While the name of Alzheimer's disease was coined for more than 100 years, and there was still no cure. The reason it may be because Alzheimer's disease is a very complicated disorders or a group of disorders, but with similar pathological feature, including a shrinkage of the cortices, enlargement of lateral ventricles, and the shrinkage of the hippocampus as well as entorhinal cortex. The two disease-defining pathological features are extracellular A-beta plaques and intracellular tau or tangles. So the majority drug development are targeting on the elimination of those two brain garbage, as well as the other neuronal transmitter regulators. However, the medical benefit of those counter drugs were reduced from major to low. In the past 20 years, there have been so many drug candidates in clinical trial, but almost all failed. So last year, Aduhelm was approved, and this year, another anti-A-beta compound was approved. But this one was a big controversial. And this year, that one, which was released 1 week ago, can reduce the progress 20%. So not so dramatic, and we still need to working on new mechanisms and new drug development. And our angle is to understand Alzheimer's disease via an aging angle. The rationale is because aging is the greatest risk factor of Alzheimer's disease. And we checked the different pathologies of Alzheimer's disease using the brain tissues we got from Harvard Brain Bank. And we finally focused on mitochondria because we continue to saw, like for Alzheimer's disease, there is an accumulated damaged mitochondria in AD hippocampal brain tissues compared with cognitive normal people. And we wondered whether this could be a neglected reason. So why mitochondria? Because the human brain constitutes 2% of body weight, but it can consume up to 30% of the energy. And we need a huge amount of mitochondria at high quality to do the work. And mitochondria are the engine of the human brain. So we propose that when we are young, we have a healthy engine, which enables this garbage clearance trunk to work efficiently, and our brain is sharp and clean. However, when we are getting older, when the worn out of the engine because of damaged mitochondria, which could result of the dysfunction of the garbage recycling truck. And with this in mind, we propose that in addition to tau and A-beta, that accumulation of damaged mitochondria could be a risk factor if we don't see an independent cause of Alzheimer's disease. And how we test this hypothesis, because we can genetically and pharmacologically reinstall impaired mitophagy. If this could inhibit or at least slow down the disease progression, then it highlights the importance of this pathway, which will open doors for new drug development targeting on mitophagy induction, for example, to using NAD precursors as we reported before. So with this in mind, we quantified mitophagy basal level, and indeed, Alzheimer's disease had a 50% to 60% reduction of mitophagy compared to sex and age matched cognitive normal controls. And speaking of molecular mechanisms, many mitophagy-related proteins like ULK1, as I showed you, were reduced in the patients. the phosphorylation of ULK1 at 555 was essential for the initiation of autophagy and mitophagy. However, the levels were dramatically low in Alzheimer's disease. So based on those data and other data in human brain tissue, AD, iPSC-derived neurons, 2 AD mouse model, APPPS1 and triple TG, as well as different ADC Adams models, with the data which unambiguously enable us to propose a hypothesis that in aging genetic and non-genetic factors, which drive the impaired mitophagy, which could contribute to accumulation of damaged mitochondria. As I mentioned, this pathway itself may not sufficient to cause Alzheimer's disease. We still need other factors that contribute to tau and A beta. And there's intertwined interactions among them, which could contribute to neuronal dysfunction and even death, as well as the impairment of the garbage clearance. For example, at a cellular level, one cell type called microglia, they are important to engulf extracellular A beta plaques. However, in Alzheimer's disease, this activity is reduced. And this activation of microglia also increases inflammation via upregulation of the NLRP3 caspase-1 signaling pathway. It may all finally contribute to an AD-like brain and memory loss. So how we show impaired mitophagy is an important risk factor of Alzheimer's disease, if not an independent or sufficient cause. Because when we genetically and pharmacologically reinstall mitophagy, which increases mitochondrial homeostasis, increase the function and survival of neurons, improve the phagocytotic capacity of microglia to reduce A beta plaques and reduce inflammation, as well to reduce A beta and tau intracellularly as well as extracellularly. And this may contribute to a healthy brain. After the publication of our paper, Nature Review Drug Discovery made a commentary. It highlights the importance of the turning up mitophagy, because if you see Alzheimer's disease is a group of disorders, there are many risk factors or pathological features. And the counter strategy, if targeting on one, may not be sufficient. However, if we targeting on mitophagy induction, which can alleviate many key features of Alzheimer's disease, like A beta, tau, inflammation, energy deprivation, and increased survival of the neurons. So if one drug targeting on many steps, many causes of Alzheimer's disease, we may have likely have better clinical benefits. So then my project number 3 to show is to understand what is the molecular mechanism of impaired autophagy and mitophagy in Alzheimer's disease. And if you can see, this is the pathways of autophagy. And ULK1 and AMPK is reduced in Alzheimer's disease. And many other proteins like PIC-ALM, beclin-1, PI3P, MBRA1 is either mutated or reduced in this in Alzheimer's disease in this step. PIC-ALM here, and lysosome colleagues also showed reduced function and levels of cathepsin B and D in Alzheimer's disease. This also applies for mitophagy, so ULK1, AMPK, and many mitophagy proteins are reduced in Alzheimer's disease in different steps. And our focus is on ULK1. If you see here are the protein clusters between mitophagy pathway and Alzheimer's disease. Many proteins are clustered together of these physical interactions, including ULK1. As I showed you, our previous data showing ULK1 is reduced in Alzheimer's disease. Its activity is also impaired. So our unpublished result showed that ULK1 protein is reduced in serum samples in Alzheimer's disease. as well as in CSF samples in Alzheimer's disease. To investigate whether reduced ULK1 is a risk factor of Alzheimer's disease or not, we generate the ULK1 overexpressed mouse model and crossed with an 5XAD mice. And our data show here the red ones. So we use Morris water maze for learning and memory experiments. And the red ones are the disease model. They can learn very slow, while the dark one is the wild type. They can learn faster. And you can see with the ULK1 overexpression, we saw increased learning capacity. So the third paper is in preparation. And right now I'm going to the last part of my talk. Because based on our mechanism, we wanted to do drug development. We have been using artificial intelligence led by 2 talented students, including former postdoc, Dr. Chen Nongxie, and DPhil student, Alice Rich, in collaboration with our colleagues. AI has been used in many fields, including in medical field, like biomarker discovery, clinical trial design and analysis, as well as drug screening, longevity promotion. In this book chapter we published recently, we summarize such functions. In this paper, we have built an algorithm to quickly screen potential mitophagy inducers. So the method we used was a simple machine learning approach, which involved vector representations of molecular structures, pharmacophore fingerprinting, and conformer fingerprinting. And this enabled us to make an algorithm for the screening of mitophagy inducers. Due to time concern, I want to give you a summary of this paper, which we published very early this year. So we submitted the database with 17 to 19 known mitophagy inducers with translational potential. We excluded those mitochondrial toxicant because they are not translational applicable. And then we built our algorithm. And with this algorithm, we went access to a virtual library from University of McCall. This library contains over 3,000 molecules identified from traditional Chinese medicine, which has been used to treat Alzheimer's disease for thousands of years, or memory loss for thousands of years. And if we give like cutoff score 0.75 with 1 as the best, we have top 18 compounds as AI highly top scored. And then we went to cell culture-based mitophagy assay using the HeLa-based mitochema compounds with 10 micromolar as cutoff. And 7 to 8 compounds were able to fulfill this requirement. Why I say 7 to 8? Because 2 share very similar structure. And then we asked whether those top 8 compounds were able to induce mitophagy in neurons, as well to improve memory in a simple animal model, C. elegans. And 2 compounds fulfill the 2 criteria. And then we subjected 2 compounds to the mice by using the 3xTG80 mice, which harboring both A beta and tau pathology. And very luckily, those 2 compounds were able to induce mitophagy in the AD mouse brain, as well to reduce memory loss. And with supportive results, we have filed a patent for R, which is pending approval. So I want to just show you one piece of the result on this AI-based drug development. So if maybe somebody do not know how to do the memory in mice, I want to share with you. It is use the Morris water maze. The understanding is when you put a mouse in the water, it needs to find a way to escape. And if it's a milky water, we put a platform underneath of the water. The first time the mouse needs like a few minutes to find the platform. If it is able to remember, then after 6 to 8 times, it can find the target in a very short time. And you can see this is our 3xTG mice. even after 6 times of training, you still cannot find the target, just randomly explore. However, for this, the same lead and with our lead compound treatment from 12 months for 2 months, and you can see this mouse was able to remember which was in this region, rather than like this one, oh, just missed it. And we definitely had like 2-way, like blinded way for our experiments. And with the quantified results, you can see with our lead compound K and R treatment, we saw dramatic increase in learning and also memory. Speaking of molecular mechanism, the compounds were able to induce mitophagy, reduce A-beta plaques, as well as to reduce different p-tau sites. For example, 181, this is a 3-FTG AD vehicle group with a K treatment, with R treatment. It can reduce many tau sites. So here I wanted to give you a summary of today's talk. I showed you DNA repair deficiency induces mitochondrial dysfunction via NAD plus depletion. The NAD mitophagy axis plays a pivotal role in neuronal resilience and is compromised in Alzheimer's disease. Defective mitophagy is a risk factor of AD, while mitophagy is a drug-able target. ULK1 is reduced in Alzheimer's disease, while ULK1 augmentation forestalls AD pathogenesis. We use artificial intelligence, which has been validated as efficient and useful way to identify novel mitophagy inducers. And we have identified 2 mitophagy inducers with 1 with patent submitted. So when we are working on aging and on Alzheimer's disease, and we noticed that aging is a cause of many neurodegeneration disorders. So my postdoc, Sophia Lotra from Aarhus University in Denmark, she wanted to explain why aging is a shared risk factor of different neurodegenerative disorders. And we propose this NAD, impaired NAD mitophagy access as a shared mechanism for both Alzheimer's disease, Parkinson's disease, Huntington, and ALS. So I just want to share with you, impaired mitophagy is a necessary but not sufficient cause of different neurodegenerative disorders. And this is a feature to explain the aging as a role. It is not specific to any of those disorders. So because of the importance of impaired autophagy in aging and neurodegeneration, we, with our colleagues like Linda Patrick, Guido Cromer, John Lebetier, David Rubinstein, that we propose compromised autophagy is an independent hallmark of aging. We have been very lucky to be involved in clinical trials using NAD for premature aging disorders, as well as for neurodegenerative disorders in both United States and Norway. As I talked to Carmen in the beginning of my talk, so in Norway, we have built this Norwegian Center, Norwegian National Anti-Alzheimer's Disease Network, focusing on consolidate the resources for anti-Alzheimer's disease research with our board members are here. I hope you still remember the question I asked, how do you foresee your health in your 90s? I hope we won't be so pessimistic, but we'll be more optimistic to have a strong physical health, as well as a sharp brain to enable us to have a happy and longer life. And, you know, I think for people who are sitting here, I want to share with you how to, you know, any secret and to promote a healthy longevity. One secret is NAD plus is available in both milk and beer. So you could consider to have a glass of milk in the morning and a glass of beer in the evening. However, life is not that so simple. The reason is because it's very low in milk and beer, and you need to drink a huge amount of beer to qualify this. That is why there are a lot of drugs available. And finally, I want to thank my team members, especially Dr. Chenlong Xie, who was the leader for this AI project and now an associate professor in Wenzhou Medical University in China. And my former postdoc, Yaya Arman, now an editor in Nature Aging. And my collaborators, as well as my major funding and my conflict of interest. And I thank you, Carmen and George, for this opportunity. I'm happy to take any questions.
Thank you very much, Evandro. A very, very nice talk. If anyone have a question here, I will check the YouTube if someone have a question. It's a little bit delayed. Gerardo, go ahead, please.
Wonderful talk, wonderful. The mechanism leading to mitophagy because of the depletion of NAD plus can participate in other types of autophagy. For example, autophagy of other organelles or the plasma membrane.
Yes, I think autophagy can happen in different tissues. For example, when we're giving exercise, starvation, or NAD treatment, it can cause autophagy induction in different tissues. So currently, the general understanding is there is an age-dependent reduction of autophagy, but that is tissue-specific. Some tissues are more easily to be affected by age-dependent reduction of autophagy. Some are not. This also applies to the drugs and any behaviors.
And I would like also to ask you if the mechanism, the pathological mechanism that you described for Alzheimer disease can also participate in other types of dementia, like the frontotemporal dementia, which is very abundant.
Yeah, so I hope you recall that slide I showed you, which we published in Cell Metabolism in 2019. We propose impaired NAD plus mitophagy pathway as a reason of why aging is a contributor of different premature of different neurodegenerative disorders. In view of the similarity of frontotemporal dementia to Alzheimer's disease to ALS, I think there could be a possibility. And indeed, colleagues in the field has been shown there is an impaired autophagy and mitophagy in frontotemporal dementia.
Nice. Finally, in your drug library you tested, is there any cardiotonic steroid included?
Oh, would you repeat again which type of compounds?
Cardiotonic steroid, like Wabain, for example? Do you remember?
I think it must be, because we have a total of 3,200 molecules inside. Because I'm not a biochemist, so I didn't look them one by one. But if you're interested, I can ask my colleagues. And if you can send me an email, if you're interested, I can ask my colleague to double check.
Okay, thank you. I will.
Thank you, Uben.
Okay, we have a couple of questions from our people that is in YouTube. I will give you some of them. Some of them are asked, could you comment on the effect of NAD on aging glia?
NAD on microglia?
Glia, aging glia, glial cells.
Okay. Yes. So in one of our very early studies, we have tracked the effect of NAD, whether it's only specific to neurons or can be also applied to microglia and astrocytes. And the answer is NAD can also increase mitophagy in astrocytes and microglia. We had some of the data in this Nature Neuroscience paper in 2019, showing the increased mitophagy in microglia is important for phagocytotic capacity of microglia to reduce A-beta plaques as well to reduce inflammation.
Okay, thank you very much. I have another, there is another question here. What about the reports of pathological role of truncated tau fragments are parking on promoting mitophagy neuron death?
Would you please repeat it again?
Yes, I will do it.
The
What about the reports of pathological role of truncated tau fragments and parking on promoting mitophagy neuron death?
Okay. Yeah, so I think there are two things. So definitely if you put fragmented tau or A-beta into the cell culture, they can also induce autophagy and mitophagy because there's a cellular response to eliminate them. And our current understanding is for the APP-PS1 mutated or APOE4 background of Alzheimer's disease, they have a genetic-based reason of reduced autophagy capacity, either at the initiation of the machinery or because of the lysosome dysfunction, which caused a reduced capacity to reduce those brain garbage. And speaking of the pink one or parking, yeah, I think there was a paper published by my colleague in AMBO journal a few years ago, showing why mitophagy is impaired in Alzheimer's disease. That was because of tau can inhibit the mitochondrial localization of parking to inhibit the pink one parking pathway to reduce damaged mitochondria.
Okay, thank you very much. Another one. There is a relationship about mitochondrial autophagy decrease and NAD dysfunction with the highest prevalence of Alzheimer's disease in women?
You mean any gender-related things?
Yes.
This is actually a very interesting and important thing. My former colleague in NIA, Tyler Demarest, when he was doing his PhD in University of Maryland, he published a series of papers showing like why men are more easier to get the brain damage than the women. So at least the studies in animals showing under the similar amount of stress, female animals are more easier to initiate these responses as well to increase mitophagy as a neuronal protective way, while men are more stubborn in this way. That is why in all of our country experiments, we include both genders in our experiments when it is like available.
Okay, any other question from here in Zoom? Jorge, adelante.
Professor Funk, thank you very much for this wonderful and outstanding talk. So the dilemma of causality. So to what extent is NAD decrease a cause or a consequence of aging? Is there any possibility of additional molecular mechanisms upstream NAD depletion?
Yeah, this is a very, very important question. So the importance of NAD in aging was not in the limelight until the recent 10 to 15 years. So people are interested in what are the molecular mechanisms of NAD reduction. I think there are a couple of reasons. The first one is when we are getting older, for example, you have more chances to expose to the sunshine. You have more DNA damage. When you have more DNA damage, which increases pyrylation, which uses NAD to generate the tree branch structure to enable DNA repair. And this causes reduction of NAD. And the second thing is a group of protein called CD38. It participates in inflammation. So when we are getting older, when you have higher inflammation due to different reasons, and this CD38 also consumes a huge amount of NAD plus. And definitely, I think there are many other molecular mechanisms people in this field are working on. Thank you, George. Yeah.
Thank you.
We have another question from our public. What is the impact of your molecule R on amyloid beta and tau aggregation in AD mouse model that you are using?
Yes. So due to the time concern, I didn't show you the Western blotting data, the immunohistochemistry data, but we published this in Nature Biomedical Engineering early this year. A long story in short. So first, we showed that our compound K and R were able to reduce intracellular A-beta peptide 1 to 42, as well as the ratio between 142 and 1 to 40, as well as extracellular A-beta plaques. So for the intracellular, it could be caused by reduced production, as well as increased consumption. For the extracellular A-beta plaques, it is likely to be caused by increased phagocytosis by microglia. So microglia can eat those brain garbage. And for tau, as I showed you, those compounds are able to reduce tau phosphorylation. When the phosphorylated tau is reduced, we presume there's also a reduced tau tangles. When we use immunohistochemistry, we indeed saw the tau tangles were reduced in the 3-XTG AD mouse brain.
Okay, and we have another question. We have many questions. Another question. Are the mitochondria of the synaptic terminals less affected by autophagy than those of the somatodendritic compartments in ID alterated neurons?
Yeah, this is, I think, extremely important thing we need to pay attention, because mitophagy in normal cells like HeLa cells or fibroblasts are totally different from that case in neurons. As Carmen, you are an expert on this. So because neurons, we have the dendrites and very long axons. This needs the mitochondria to be transferred from the soma to the distal region of the axon. And then in the synapse region, which you have the distal region of the axon, as well as, so these all need a lot of mitochondria there. So there have been intensive studies to show the instance of mitophagy. The long story in short is mitophagy is majorly happened in the soma region, in the soma region of the neurons, but it is also frequently happened and also important in the distal regions of the axon, as well as in the dendrites.
Okay, thank you very much. Does anyone have an additional question here from the Zoom? Because from the, I have another question. It's one. It's what about exercise?
Yeah.
And autophagy? I'm sure you have explored there and the impact that is having in all this process.
You mean the role of exercise in this?
Exercise, running, exercise.
Exercise, yes. So we have two students in the lab. I think Helin Wang, so she's actually here. So she and a postdoc, Janet, they are working on the combination of NAD plus exercise and see any additive or synergistic effects in reduced memory loss. And we saw indeed, at least in C. elegans, we saw additive benefits. And definitely that is why we have made an appointment with Henrietta van Praag on the 10th of like next Monday to get more comments from her. Yeah, definitely want to ask you.
Very nice. Yeah, this is very, very nice. Another question. I think we don't have more questions. Thank you very much, Evandro, for the excellent talk. Hopefully we can have you here in Mexico with us, giving us a talk, and we have the opportunity to meet a lot of people that is interested in aging and Alzheimer's disease here in our institution. It has been a great pleasure to have you here.
Definitely. Thank you guys so much for this opportunity, and let's keep in touch.
Yeah.
See you. Bye-bye.
Bye-bye.
Yeah. See you, Gloria. Yeah.